Hello, this is short oral discussion presentation number 632, Dynamics of hexokinase-2 linked glycolytic overload mediating endothelial cell dysfunction in high glucose concentration in vitro. I'm Paul Thornelli. Metabolic dysfunction in endothelial cells in hyperglycemia is initiated by glucose-induced stabilization of hexokinase-2 to proteolysis. There are multiple pathways of metabolic dysfunction in hyperglycemia: hexosamine pathway, PKC activation, and dicarbonyl stress. The first enzyme metabolizing glucose is hexokinase. There are hexokinases 1 and 2 in endothelial cells. Both are saturated by glucose even at normal glycemia. In hyperglycemia, it's a unique stabilization of hexokinase-2 to proteolysis that gives rise to increased glucose consumption and all metabolic dysfunction. High glucose-induced hexokinase-2 linked responses include increased flux of glucose metabolism without transcriptional increase of glycolytic enzymes, producing a wave of increased glycolytic intermediates. This stimulates hexosamine, PKC, and dicarbonyl stress pathways. Increased glucose 6-phosphate produces detachment of hexokinase-2 from mitochondria, mitochondrial membrane polarization, and increased reactive oxygen species ROS production. These effects are reversible and preventable by GLO1 inducer transresveratrol hesperidin. The aims of this study: to investigate the dependence of hexokinase-2 linked glycolytic overload on glucose concentration and duration of exposure to high glucose concentration, to explore the link of increased hexokinase-2 with increased flux of consumption of glucose driving metabolic dysfunction in hyperglycemia, to study the rate of return of hexokinase-2 to basal levels with switch to normal glucose concentration, to study the response of hexokinase-2 linked glycolytic overload to glyoxylase 1 inducer transresveratrol hesperidin combination. The experimental model is human aortal endothelial cells in primary culture incubated from 0.5 to 6 days with baseline glucose concentration 4.1 to 20 millimolar glucose with and without 5 micromolar GLO1 inducer. The results: in model hyperglycemia, 20 millimolar glucose, hexokinase-2 protein is increased at 12 hours and remains maximally increased thereafter. Hexokinase-2 protein increases progressively with glucose concentration in the diabetic range, increasing maximally by 37% at 20 millimolar glucose. Increased abundance of hexokinase-2 protein at 48 hours correlated positively with baseline glucose concentration and flux of glucose consumption. Increased hexokinase-2 protein abundance in high glucose concentration is reversed by 2-day switch to low glucose concentration. GLO1 inducer transresveratrol hesperidin corrects increased hexokinase-2 protein, increased glucose consumption, and all metabolic dysfunction. In conclusion, hexokinase-2 abundance increases with increased glucose concentration in the clinical range in diabetes in human aortal endothelial cells in vitro. Increased hexokinase-2 without concurrent increase in glycolytic enzymes produces a wave of increased glycolytic intermediates in early stage glycolysis driving metabolic dysfunction. The slow return of high hexokinase-2 abundance to basal levels after a period of high glucose concentration exposure suggests metabolic dysfunction is carried over from increased glucose concentration in the postprandial phase to the fasting phase. Finally, GLO1 inducer prevents this by normalizing hexokinase-2 protein and may provide effective treatment of endothelial dysfunction linked to hyperglycemia and diabetes. Finally, I'd like to thank Dr. Mingzhang Zhu, Diabetes Research Center, QBRI, HBKU, Qatar, working with me. We thank our collaborator, Professor Naila Rabbani, College of Medicine, Qatar University, Qatar. We thank the Qatar Foundation and Qatar University for funding our research, and we thank you.